Please go ahead.
Thanks for a wonderful talk. For those cardiac safety screening, I know that for iPSC-derived cardiac cells are electrophysiologically or metabolically not mature, especially like when you're considering calcium handling. I think we're still limited with a lot of these true EP physiology to represent the adult cardiomyocyte. So I wonder how we can be mindful about those when we try to call it cardiotoxic or the safe drug.
And you get pretty close to it. Oh, I think we're turning them on still.
Well, I guess I'm not sure which of us panel members that was directed to, but I share your understanding of the overall maturity or immaturity of some of these model systems. I mean, a lot of cardiac tissue engineering efforts have gone into improving maturation or the ability to recapitulate more adult phenotypes. And that's why I think these can be used in a way to screen. But in general, if you think about the SEPA approach that I talked about before, it really combines multiple streams of data to overall understanding the potential arrhythmogenic risk. And maybe Amir can comment on what he's seen as part of his modeling too, based on how he's designed the systems.
Why don't you step up here?
Yeah, is this better? So a very valid point, and we have one of the world experts in differentiation of cardiomyocytes in the room, so I would like to get his perspective as well. The question was about the validity of the cardiac models that people are using, differentiating from iPSCs and the immaturity that is part of the game. I'm addressing it, but if there's any comments, it's going to be privileged to hear from you as well. I think the main assignments from our side is first of all to understand that the models that we're going to be using, they're not going to be perfect. In some cases, they're going to be closer to capturing reality. In other cases, they're going to be completely far off. Like I mentioned, the work that we've done on the liver differentiation, when we started, there were protocols out there. People were reporting that they're actually functional, but eventually we saw that they were not. Same with the cardiac model. The idea from our side, at least the approach that we're thinking, we're not trying to be experts in differentiation of cardiomyocytes. There are people that are doing it better than us probably. The idea is to use the best acceptable protocol that we can use in order to capture the right responses, taking into account that we need a few parameters to have an overall estimation of what's the reality. One of the things that we discovered is that sometimes even the imperfect model that you're using, if there's an underlying genetic background that's causing an effect, you can actually capture it even if the model is not 100% representing the mature cardiac function. I'm sure that there are going to be cases that it's going to be even more difficult to come across specific responses. Not all conditions, cardiac conditions, are similar to each other. So some of them will probably be more successful in capturing, others will not. But we need to start somewhere. We need to have tools to do the best prediction on the individualized level and work from there, continuously identifying the gaps and filling them as much as we can as we move. Assuming that we can fix everything right from the beginning is going to be too presumptuous, so I wouldn't go there.
All right, any other comments on the first question? No. All right, well, let's next question.
It's on. Yep, there we go. Especially with regard to these large data sets and databases that are going to power the AI models, the in silico models, how do we deal with the language imprecision? And I'm accusing myself of this. I call my brain chip a brain chip. It's not really a brain. I call my liver chip a liver chip. But does that liver chip contain hepatocytes and cholangiocytes? Does it contain an immune component? Is it a Kupffer cell or is it a THP1 cell? Those details do matter, but when you're talking about things, you also don't want to get so bogged down in them that you can't say something significant about the liver. But I do know that just with dealing with databases, if you don't use the same terminology, you don't get the same answers. Query the system.
Yeah, who'd like to take a first answer here?
It's working now?
Yes.
I can take a first pass at this. I think you're describing reality with all the complexities that we need to deal with. From my perspective, the most important part is translation, looking into things that we do at the lab that are definitely just a fraction of reality. Similar with the prediction and what we're doing on the AI side. It can capture reality, but only to a certain extent. The idea is to continuously move forward to make sure that we're mapping the gaps and filling them. One of the things, and I tried to paint it out at the beginning, hopefully it was captured, we found out that one of the most important parts is the quality of data. We've seen multiple analyses being done, and I really appreciate the fact that people want to use all data as much as possible. People spent years, money, and energy to create this. But we really need to be vigilant about how we handle the data and the type of data that we're using for the analysis. So eventually, I think this is the starting point. We need to make sure that we create good, qualified data sets. We train our models based on that. And moving from there, we can start really mapping out the gaps that we have. Otherwise, there's too many variants in this equation, and it's going to be impossible to answer it.
Great, thank you. Others? Okay, let's move on to the next question. Go ahead, Bino.
Okay, Bino Friedman, University of Washington. Really interesting session, very cutting edge stuff. My question was really for Dr. Tong, but can be maybe addressed by multiple groups. So some of the data you showed with the animal GAN, for instance, I just wanted to clarify, it seems that you could take a compound structure potentially and input it into the system and make a prediction based on that alone that could be superior to actual experimentation results. So I think that's a very intriguing idea, but I'm also sort of a little skeptical that the state of our, abilities or the abilities of AI to really do this, based purely on sort of a Bayesian approach would be sufficient to avoid things falling through the cracks. So I'm curious, where you think that state of the art is, because it would be much faster and could be envisioned as something that could become really part of the standard pipeline, if true. But, skipping experimentation entirely would seem to be a little risky if we were to go into humans.
Well, thank you for the questions. And regarding the animal GAN, we are not only just take a chemical structure alone, and we also add the dose information as well as the treatment duration. So this is how the model was trained, and based on 3 sets of the information: the chemical themselves, dose, and the treatment duration. So when a new compound's coming in, so you have to ask a question at a certain dose, maybe high dose or low dose, at a certain treatment time, for example, 28 days, and what is the outcome going to look like. So this is what we are focusing on for the animal GAN. And in terms of what is the risk to apply these models and in the real world setting, and as a context of the use, so we are envisioning this kind of the models are going to be used in the preclinical drug screening, as well as to make it, or maybe just reduce the animal use. Instead of you use 10 animals to do the studies, you might be able to use these models to generate the results and then being confirmed with a much less number of the animals to use. Now, what is the potential risk? Now I have to say, and the models we developed only based on very small number of the compounds. For the animal GAN, we are involved close to 140 drugs. And so we are a little bit nervous about 140 drugs. It's not given broad coverage of the chemistry space. So what we did is we did a quite extensive validations, internal and external, you name it. It's all the standard procedure we use in the AI area. But we did extrapolate this result to the 355 drugs, and we gain a lot of the confidence, and then we work on 28 preclinical drugs, which so far we still don't know what this drug it is, because we work with the AbbVie, we give the program to them, so they generate the informations. So now we are working with the BI as well as the GSK, and to test their preclinical drug candidates. So I hopefully after testing on the various drug candidates from the different pharmaceutical company, and we will be able to gain not only just confidence, but also the applicability domain when this model is going to be used and with a confidence.
Thanks, Wade.
Okay, thanks.
Follow-up or no?
No follow-up. That was great. Thank you.
If I can have a follow-up. So Wade, thank you so much for bringing this topic of trying to be very careful in messaging and being also very careful with applicability domain and and not overpromise. While Dr. Hartung is not here to answer this question, maybe I can get other panelists to maybe talk about this seeming disconnect about 100, 200, 300 compounds. We're trying to build this slowly versus just throwing the kitchen sink and getting 110% accuracy. So how do we message this correctly to the end users, both the FDA reviewers and the sponsors and also the research community? Because if one just, comes to this meeting only and listens to this, they say, well, my job is done. I just pack up and go home. There's nothing new for me to contribute. So maybe we can get the panelists to talk a little bit about, where the middle ground maybe is between the 100 to 300 and 26 billion compounds, and how do the end users actually process this information?
Thanks, Yvonne. Who would like to take a first pass right now?
I can take it. So I'm going back to the comment that I made before. I think the game of numbers is the wrong game. Building confidence is the right approach. For now, for me, the most disturbing part is making sure that there's the right translation in everything that's being done. If someone scans hundreds, thousands of drugs moving forward, but there's no translation, then basically there's nothing there. When I'm talking about translation, I'm talking more about the personalized response. I talked about it that the average response is great as a starting point, but eventually I think we need to get to see how an individual is actually going to respond to this drug. This is a huge undertaking. I think this is the actual assignment. Continuously improving the systems and saying that one can get to 91, 92% versus someone else that can do 85, I think we passed this hurdle. We're not there. This is not the game now. The game is to make all these platforms usable to the point that we can really affect human lives. And affecting it is not by saying that we can have better, safer drugs for the entire population, but we can tailor it for specific subpopulations and for individuals as much as possible. This is maybe in the long run. This is definitely not something that's going to happen tomorrow morning. But starting to really look at the individualized response, this is the name of the game now, from my perspective. And just to summarize it for your specific question, Yvonne, I think going into the loophole of saying what end user, which approach each one is going to take to kind of compete with the other parties, saying that I can have better accuracy, or I can have a few more drugs, we should really step out of this game. I think there's great minds in this room and the entire community. As long as we have the focus on the actual prize of where we want to get, and to whatever extent we can join forces or at least support the acquiring of knowledge as we move forward, this is going to be the best for everyone. Hope it addresses the concern.
Thanks. Wade, did you have a perspective you'd like to add as well?
Yes, I would like to add, first of all, I really agree. And if we just focus on the number of the drugs or number of the compounds, we needed to develop a model. Maybe this is just too simplified and this complex the questions. And when we look at the data, we normally look at the data quality, and this has been highlighted in the previous presentations. We also look at the variations, we look at the prevalence. So there is a number of the sort of the measurement of the data determines how many data, how many and the drugs you probably needed. But also, and I often struggling with a lot of the models, particularly in the drug-induced liver injury area, is we mainly work on the marketed drugs. So the marketed drugs, in principle, is safe when came out from the market, and only very small number of the people were suffered from the liver injury. And so when we decide whether this drug causes liver injury or not, it's a subjective matter, right? And let's say you got 1 people died compared to 10 people and have an ALT elevation. How are you going to make a judgment whether this drug is causing, 2 drugs, whether, which one caused liver injury or not? And so the annotation themselves has a lot of the room for improvement to themselves. And then you can ask a model to give a better prediction for the endpoint, which the annotation already and sort of the varies. So we really need to take this all these issues into the consideration. The bottom line is, if you develop the models, we need to look at those models, apply this model in the various and the scenarios, and to gain the confidence on when and where these models may be able to contribute to the drug safety assessment, for in our case for the DILI. And as a job, I think it's Bob Job was saying, all the models are wrong and some may useful. I feel this is the mentality we need to carry through when we look at the model and trying to find the sweet spot that the model can be useful.
Okay, thank you, Wade. And I think we have time for one more question right now, and then we're going to move into the first break for the day.
Okay, great. Maybe last question. I resonate with Dr. Ishida's the idea about inline monitoring. I really respect that's an excellent idea. I was just wondering, do you have a plan to really put all different sensor in the system? And currently, all this MPS system is just microfluidic and cell culture. There is no real sensor. But if there is a sensor, I see many people put the electrode, including myself. But then if you measure cell response with the electrode, there's inflammation, and people completely ignore this, and then they get the signal from astrocyte, not from the neuron. Then you have to think about what is the best way to collect this data and so on. It's not only electrical sensor, but also even other optical biosensor. Using the virus to modify optogenetics. Is that really reasonable game to really find the best drug? If we use invasive method, you have to think about what is your plan in inline monitoring, non-invasively.
So I thank you very much for the question, and I just have the plan to incorporate, for example, the glucose sensing or the lactate measuring, and also I think the cell morphology helps us to judge if a cell is healthy or not. So not so many things I'm going to incorporate to the MPS because it costs a lot. And also considering the throughput of the MPS, I think it is not realistic to incorporate many sensing. So, but depending on the cell type, for example, as you told that the nerve system, maybe the electrode is very important to measure the cell functionality. So that depends on the cell types, I think. And we are now start discussing with the Japanese CROs what kind of data they are required or the thinking to important for the documentation. Yeah. So it's just starting the discussion now. Yes.
Thank you.
Well, thank you very much, everyone, for your questions this morning, as well as to the presenters. And we're going to take